The next item on the agenda today is an information item on the biological mechanism acting at low doses of radiation. This is outlined in CMD 17 M46 and M46.A. I understand, Mr. Rinker, you still have the floor. Over to you.
Thank you, Mr. President and members of the Commission. My name is Michael Rinker, Director General of the Directorate of Environmental and Radiation Protection and Assessment. I am accompanied by staff members of the Health Science and Environmental Compliance Division, Ms. Julie Burt and Dr. Julie LeBlanc, both Radiation Health Science Officers in that division, as well as Dr. Rachel Lane and several other subject matter experts to help address any questions. We are also accompanied by Dr. Patsy Thompson, who serves as the CNSC's technical advisor, as well as Vice Chair of the United Nations Scientific Committee on the Effects of Atomic Radiation, or UNSCEAR. We are here today to present CMD 17 M46 in response to a request from the Commission following a public meeting in Ottawa on April 6, 2016. The Commission requested that staff present the current science on the effects of low doses of radiation. I will ask Dr. LeBlanc to begin the staff's presentation.
Thank you. For the record, my name is Julie LeBlanc. As mentioned, I am a Radiation and Health Sciences Officer. Before I begin with the presentation, I'd like to draw your attention to the picture of the cell on this slide. It is here as a reminder that today's presentation will be focused on radiation biology, the field of study that looks at the effects of radiation on living things. Typically, experiments in this field are performed on cells, parts of the cell, or on whole animals. Radiation biology complements epidemiology. The latter is the study of the factors affecting health and illness of human populations and how disease is distributed. Until recently, epidemiological studies could not provide firm conclusions as to the health effects from the low doses of radiation, nor can they offer any explanation of what is going on inside the human body following radiation exposure. Radiation biology experiments are useful as they can offer a biological basis for what occurs within the low dose range. To understand the effects of radiation, we will first review some fundamental concepts in radiation biology. Within the radiation protection framework, there are three different but related types of dose often discussed. The first, on the top of the screen, the absorbed dose is expressed in a unit called the gray, but more commonly referred to as milligray, which is 1,000 times smaller. It describes the energy deposited by radiation in a kilogram of substance like tissue. When discussing human health studies or radiation biology studies of cells or animals, this is a unit typically being used as it is a physical unit. The next two units are radiation protection quantities. This means that judgment has entered the equation since both terms, including weighting factors, grounded in science but determined by scientific committees. The equivalent dose expressed in sieverts, or more commonly millisieverts, is the absorbed dose weighted for the different types of radiation. This weighting factor accounts for the fact that for the same dose, some types of radiation are more biologically damaging than others. The example on the middle of the screen shows that alpha radiation is 20 times more biologically damaging than both beta or gamma radiation for the same dose. At the bottom of the screen, the effective dose, also expressed in millisievert, is the equivalent dose weighted for the sensitivity to radiation of the organ or tissue exposed. Some tissues and organs are considered to be more sensitive to radiation. That is, radiation can be more harmful to certain tissues or organs. This occurs because the cells that make up those tissues divide more frequently. You can see in the diagram, three different examples of weighting factors are listed. One for the testes, one for the lungs, and another for the bladder, demonstrating that the testes have a larger weighting factor and are thus considered to be more sensitive to radiation. The figure on this slide is providing context for the term low dose. The gray box symbolizes 100 millisievert. This is the value we tried to limit our review of the literature for the purpose of the CMD and today's presentation. At times, greater doses were considered, but our work focused on radiation biology experiments where the doses were in the low dose range, that is 100 millisievert or less. To put this value in context, we can compare the gray box to several other values. First, the green box represents the nuclear energy worker dose limit of 50 millisievert in 1 year. The 100 millisievert over 5 years component of the dose limit is not shown on this figure. Second, the blue box represents the annual average natural background in Canada of 1.8 millisievert. Third, the orange box symbolizes the annual public dose limit of 1 millisievert. Workers typically receive a small fraction of the CNSC dose limits year after year. The doses discussed throughout this presentation, which are 100 millisievert and below, are particularly relevant as they are in line with what nuclear energy workers actually receive over their lifetime. The doses to members of the public are 100 to 1,000 times lower than the CNSC public dose limits and are thus relevant to today's discussion as they are significantly lower than the doses used in experimental radiation biology studies. Another concept to understand before we move on to the biological effects of radiation is radiation quality. Radiation quality is a measure of energy that is transferred from the radiation to the exposed matter or the tissue over a given distance. This is referred to as the linear energy transfer, or LET. The LET can impact the type of damage that may happen to the cell as a result of the exposure to radiation. Therefore, it is important when comparing experimental results from different studies to know which type of radiation was used. As you can see at the top of the diagram, low LET types of radiation, like beta radiation, transfer their energy to the tissue or organ in a sparsely ionizing pattern over a large distance, whereas high LET types of radiation, like alpha radiation, transfer their energy in a dense ionizing pattern over a comparatively small distance. The implication here is that high LET radiation could cause damage to the cell that is more difficult to repair than low LET radiation. Although radiation can pass through and deposit its energy in any part of the cell, nuclear DNA has classically been considered the most critical molecule because it is the set of instructions, or genetic code, that dictates how your cells will grow, divide, and function. This set of instructions is passed from cell generation to cell generation. Damage to DNA could result in an adverse biological effect, like a mutation, which in theory could increase the risk of developing cancer. After radiation exposure, the cell will mount a response to maintain the integrity of the genome, which is the sum total of an organism's DNA, to minimize any adverse effects or biological damage caused by the radiation. Many parts of the cell, like the cytoplasm, mitochondrion, and cell membrane, as shown on this image, all have important roles to play when a cell responds to radiation. The figure on this slide shows a cartoon version of DNA where the two sides of the molecule are linked and together form what is called a double helix. If radiation does deposit its energy in the nucleus of the cell where the DNA is housed, it can cause damage either through direct or indirect action. This means that radiation either interacts directly with the DNA, or the radiation interacts with atoms in the surrounding environment, creating molecules called free radicals, which in turn interact with the DNA. The important takeaway from this picture is that both direct and indirect actions of radiation can result in the same type of DNA damage. The type of DNA damage we are most interested in is called a double-strand break. As you can see in the image on this slide, a double-strand break is where both sides of the DNA double helix are severed or cut. There are many different types of DNA damage that exist, and we will not go over all of the different types, but focus on double-strand breaks as they are considered to be the most severe form of DNA damage since they are the most difficult to repair. Furthermore, DNA damage is strongly associated with the development of cancer. Double-strand breaks can occur via normal processes, cell processes, such as DNA replication or cell division. They can also occur due to environmental stressors, such as radiation, ultraviolet rays, natural or synthetic chemical agents. As stated on the previous slide, double-strand breaks can occur as a result of natural cell processes or by some environmental stressor. To put this into context, cells can sustain and repair a relatively high rate of naturally occurring DNA damage. Approximately 50,000 sites of different types of DNA damage occurs per cell per day. Approximately 10 of these naturally occurring events are double-strand breaks. Thus, if cells can adequately deal with this level of naturally occurring DNA damage, then a small number of additional radiation-induced double-strand breaks would be of little consequence for cancer risk. Again, the amount of DNA damage at a cellular level is proportional to dose. For example, a dose of 5 milligray of low LET photons or electrons would result on average in 0.25 double-strand breaks per cell per day. The fraction of a double-strand break, or the 0.25 number, is a reflection of probability that at such low doses, some cells will be traversed by radiation and develop a double-strand break, while others will not. As we've been discussing, if radiation passes through a cell causing damage, like a double-strand break, the cell will then mount a response to maintain the integrity of the genome. This response will either lead to proper repair of the damage, resulting in no increased cancer risk, or there will be misrepair or a lack of repair, leading to an increased cancer risk as indicated by the pink cell. However, an increase in the risk of cancer is just that, an increase in risk. It does not mean that a cancer will necessarily arise. This is demonstrated by the possibility that the cell highlighted in pink can either become cancerous or not, indicated by the black and blue cells respectively. What I would like for you to take away from the fundamentals of radiation biology section is that first, ionizing radiation can damage DNA, but this damage is most often repaired. Second, double-strand breaks are the most severe type of damage since it is the most difficult to repair. Third, improperly repaired damage can contribute to cancer development. And fourth, environmental exposures of radiation produce a small fraction of damage compared to what occurs in our bodies every day. I will now pass the presentation to Mrs. Julie Burt.
Thank you, Dr. Leblanc. For the record, my name is Julie Burt, and I am a Radiation and Health Sciences officer with a background in radiation biology. Next, we will discuss how epidemiological studies of people who have been exposed to radiation have helped us understand the risk of developing radiation-induced cancer. This is described by a dose-response model. A dose-response model is a theoretical model that describes how the risk of cancer varies with radiation exposure. The risks from radiation have largely been derived from epidemiological studies of the atomic bomb survivors, where there was data, typically above 100 milligray, the dose response was linear, meaning that as doses increased, so did cancer risk. However, in the low dose range, the natural incidence of the disease masked any effects that may have been caused solely by radiation. Because of this, the linear non-threshold model, shown on this diagram in red, assumes that the cancer risk at low doses can be linearly extrapolated from the risk observed at higher doses. Recent chronic low-dose epidemiological studies of nuclear energy workers have provided additional support for the LNT model. Also featured on this diagram in blue is a dose-response model called the linear quadratic model, which assumes an upward curvature between dose and cancer risk. Of note, the linear quadratic model best describes the risk of developing leukemia due to radiation exposure. Overall, the data from epidemiological studies is typically consistent with either a linear or linear quadratic dose-response model. As the nuclear regulator, we use the LNT model for radiation protection purposes. that is, to inform the choice of appropriate dose limits. The international radiation protection community understands that the LNT and linear quadratic models cannot explain all experimental data. There is epidemiological evidence supporting some nonlinear dose-response models, but this largely stems from high-dose epidemiological studies. For comparison's sake, the LNT model is shown in red on all three images on this slide. The first image on the screen describes a threshold dose-response model, shown in yellow, which assumes that there is no risk incurred until a certain dose is exceeded. For example, a threshold dose of a few thousand milligray has been observed for select cancers, like bone cancer. The middle image on this screen shows a superlinear dose-response model, shown in blue, which assumes a greater risk than predicted by the LNT model. This model best describes extremely radiosensitive patients suffering from rare genetic disorders. The third image on this screen shows a Hormetic dose-response model, shown in purple, which assumes that the risk from radiation is not only lower than predicted by the LNT model, but could in fact lead to stimulatory and beneficial effects, which at larger doses become detrimental. Although there is support for the hormesis model in animal studies, there is no human epidemiological evidence supporting this model to date. The figure on this slide is an exaggerated representation of how both low-dose radiation biology and epidemiological studies support the 5 different types of dose-response model, each with their own curve. The colors of each line are consistent with the previous 2 slides. That is, the superlinear model is in light blue, the LNT model is in red, the linear quadratic model is in dark blue, the hormesis model is in purple, and the threshold model is in yellow. In epidemiological studies, the outcome of interest is typically cancer development in people exposed to radiation. In radiation biology studies, the outcome of interest is a particular biological effect, like DNA damage, for example, which can be observed in a cell or from an animal exposed to radiation. The level of DNA damage and subsequent repair is often used to draw conclusions about cancer risk in people. What I would like for you to take away from the dose-response model section is that, first, the LNT model cannot explain all scientific data. This model is practical from a regulatory perspective and conservative from a health perspective. And 2, the international regulatory community, including the CNSC, continues to use the LNT model for regulatory purposes. The CNSC regulates based on the best available scientific data and continues to monitor developments in the scientific literature. Presently, epidemiological evidence cannot confirm any dose-response model below approximately 100 millisieverts. This is where experimental radiation biology studies of different biological mechanisms can help explain each dose-response model by providing insights as to what may be occurring in living organisms at low doses. 4 different biological mechanisms will be defined and explained in the following slides. As we've discussed earlier, when radiation passes through a cell, the cell is going to mount a response. A biological mechanism encompasses the steps between a stress response to radiation and a resulting biological effect. The effect of interest is typically DNA damage. It should be noted that each of the 4 biological mechanisms discussed can be induced by other stressors, like chemicals, for example. The reason we are discussing the different biological mechanisms is because, in theory, each one could influence the shape of the dose-response curve, and we are interested in what is going on at the cellular level in the low dose range. To review, the first biological mechanism that we will discuss today is the bystander effect. This mechanism was presented to the Commission in April of 2016. Bystander effects are radiation-like effects that are observed in unirradiated cells that are responding to communication signals from directly irradiated neighboring cells. The nature of these signals is not fully understood at this time, but reactive oxygen species are thought to play a major role. This figure shows how the ionizing radiation only interacted with one cell, highlighted by the red nucleus in panel A. And that red nucleus has sent communication signals to the pink nuclei as highlighted in panel B. As far as the cell can tell whether it has a pink or red nucleus, they will both act as if the radiation passed right through each and every pink and red nucleus, despite the fact that it only passed through the cell highlighted with a red nucleus. Importantly, the identification of the bystander effect confirms that energy does not need to be deposited in a cell to produce a biological effect, and therefore the whole cell, tissue, or organ can respond to radiation exposure. The bystander effect could be either a protective mechanism or a mechanism that leads to detriment. For example, if communication signals were to lead to the destruction of bad or unhealthy cells, this mechanism could be considered as protective. However, if communication signals were to create adverse effects in neighboring healthy cells, like inflicting DNA damage upon them, this biological mechanism could be considered as detrimental. The second biological mechanism, also discussed before the Commission in 2016, is genomic instability. Genomic instability is described by the accumulation of new genetic alterations, or changes, observed in the progeny of irradiated cells, even multiple cellular generations after the initial hit by radiation. A cellular generation is the result of a cell dividing to produce two identical daughter cells. Each new cell generation is indicated by a downward pointing arrow. As you can see in the figure on this slide, different types of changes, like mutations, shown in green, or different types of damage to whole chromosomes, called chromosomal abnormalities, shown in purple and red, can be observed multiple cell generations after the initial hit by radiation. These alterations or changes appear to be random in nature and timing. Genomic instability is a hallmark of cancer and thus considered to be a biological mechanism that solely leads to detriment. The third biological mechanism is considered to be a protective mechanism. As you can see in the figure on this slide, the adaptive response is where a small priming dose of radiation, indicated by the purple bar, serves to increase the resistance of the cell to a subsequent larger challenge dose, indicated by the blue bar, typically given several hours later. Importantly, less DNA damage, indicated by the red arrow, is observed when you combine a priming and challenge dose compared to when the challenge dose is delivered alone. The adaptive response is the best studied mechanism that supports a hormesis dose-response model. The last biological mechanism is referred to as hyperradiosensitivity increased radioresistance, or HRS-IRR. This biological mechanism is defined by an increase in sensitivity to radiation between 10 and 300 milligray, followed by an increase in radioresistance between approximately 300 and 1000 milligray. HRS-IRR is best described using the induced repair model, a model derived from the linear quadratic model. The induced repair model, shown here in blue, differs from the linear quadratic model, shown in red, as it theoretically accounts for the induction of DNA repair machinery. In other words, the induced repair model describes the HRS-IRR mechanism because it accounts for the induced radioresistance, hence its name. The molecular basis for the hyperradiosensitivity increased radioresistance phenomena is that lower doses are less capable of triggering DNA repair or other radioprotective mechanisms because there is not sufficient DNA damage to induce these mechanisms, resulting in cell death, observed as hyperradiosensitivity. And that higher doses inflict more DNA damage, subsequently triggering the repair mechanisms, resulting in an increase in cell survival, observed as increased radioresistance, which eventually reaches the cell survival rate approximated by the linear quadratic model. The mechanisms presented in this section provide an overview of the possibilities of what may be occurring at the cellular level following very low, and moderate doses of radiation. These terms are defined by the United Nations Scientific Committee on the Effects of Atomic Radiation, or UNSCEAR, are indicated on the slide, where very low doses refer to doses lower than 10 milligray, low doses refer to doses lower than 100 milligray, and moderate doses refer to doses between 100 and 1000 milligray. The figure on this slide provides a breakdown of the broad range of doses at which each individual mechanism has been observed in vitro in the literature. As you can see in red, genomic instability is observed as low as 10 milligray, but generally between 100 and even over 1000 milligray. In purple, the bystander effect has been observed as low as 5 milligray, but generally between 10 milligray and 1000 milligray. In yellow, the priming component of the adaptive response has been observed as low as 1 milligray, but generally between 1 and 200 milligray. In green, increased radioresistance, or IRR, is typically observed above 300 milligray. In blue, hyperradiosensitivity, or HRS, has been observed as low as 10 milligray, but generally below 300 milligray. For IRR and HRS, there is a gray zone for where the cell transitions from a state of sensitivity to a state of resistance. While these mechanisms have been observed over a broad range of doses, we are interested in them because they occur in the low dose range. What's more, they'll all generally occur at an optimal dose of approximately 100 milligray, demonstrating that they can be active at the same time. What I would like for you to take away from the biological mechanisms section is that low doses of ionizing radiation induces many biological changes, and these changes can be explained by the following biological mechanisms: the bystander effect, genomic instability, the adaptive response, and hyperradiosensitivity increased radioresistance. Further, to better understand the contribution of these mechanisms on human health, the study of the interaction between these mechanisms will undoubtedly be a source of important information. The relationship between the different radiation-induced biological mechanisms has been reviewed in the literature, but few studies have directly considered multiple mechanisms at once. Several key proteins active in several molecular pathways are known to be involved in all 4 of the biological mechanisms discussed in this presentation, shown in white at the top of each column. This table highlights the role of select proteins in 3 different molecular pathways important for cancer development, shown in black, listed in each row. The first row refers to DNA repair. Radiation induces DNA damage. In response, the cell activates DNA repair. The successful repair depends heavily on the levels of key proteins involved in DNA repair. The second row refers to the cell cycle regulation. In order for new cells to be generated, cells naturally go through a process of duplication and division known as the cell cycle. The regulation of the cell cycle helps facilitate the repair of any DNA damage, including radiation-induced DNA damage. The third row refers to oxidative stress. Reactive oxygen species, or ROS, are produced in the course of normal cellular processes, such as during energy production in the mitochondria. Typically, the mitochondria are equipped to reduce ROS levels when needed. However, if levels of ROS become too elevated, oxidative stress could occur, which could lead to DNA damage. Radiation can also lead to oxidative stress. Of note, while all of the biological mechanisms listed in this table have each of the molecular pathways in common, each biological mechanism may lead to the activation or inhibition of the particular molecular pathway. For example, oxidative stress is increased during the bystander effect, but decreased during the adaptive response. The similarities with regards to the shared molecular pathways discussed on the previous slide may help explain some of the other common features of the 4 biological mechanisms. Recent studies have stressed that the 4 biological mechanisms discussed here are not mutually exclusive. However, exactly how these mechanisms interact remains an important research question. Biological mechanisms could be affected by the activity of other mechanisms, perhaps even acting together or against one another. This slide highlights the need to better understand the individual biological mechanisms, but also if and how they act in concert. The answers to these questions will improve our understanding of the contribution of each biological mechanism to any potential health outcomes. So what does all of this mean? Are there any potential health implications from the biological mechanisms that we've discussed? In other words, does the radiation exposure under consideration that is causing changes at the cellular level lead to an increase, a decrease, or no change in cancer risk at the individual level? The potential health implications depend on whether or not DNA damage is successfully repaired. Persistent DNA damage could compromise the integrity of the genome. Genomic integrity, which forms the base of the figure on this slide, is the maintenance of our genetic code. It ensures proper function of not only our cells, but our tissues, our organs, and our body as a whole. The loss of genomic integrity can lead to an increase in cancer risk. As the figure on this slide demonstrates, the maintenance of genomic integrity is a delicate balance between radiosensitivity and radioresistance. Radiosensitivity could result from a combination of biological mechanisms that are driven by factors such as lack of DNA repair or an increase in oxidative stress. Radioresistance could be the result from a combination of different biological mechanisms that are driven by factors such as an increase in DNA repair or a decrease in oxidative stress. At this time, there is not enough evidence to demonstrate the impact of the biological mechanisms on radiation-induced cancer risk at low doses. More research on the interaction between all of the biological mechanisms is needed before we can determine under what circumstances the scale will tip towards radiosensitivity or radioresistance. Going forward, the careful consideration of the limitations of radiation biology studies and a better understanding of the low-dose DNA damage response will be critical in assessing radiation-induced cancer risk. It's difficult to take experimental cell studies or animal studies in an attempt to extrapolate what that means for the risk of cancer in humans. For example, careful considerations could include whether cell or tissue-specific effects observed in vitro experiments reflect the true response in the body as a whole, whether uniform animal populations who have the same age, sex, diet, and lifestyle and genetic background can represent diverse human populations, and whether experimental dose and dose rates are representative of environmental exposures. Of note, it is also possible that the different biological mechanisms impact an organism at the cellular level only and may not impact overall cancer risk. I'd like to pass the presentation back to Mr. Rinker to conclude with staff thoughts on where the science is, where it's going, and how it impacts our regulatory oversight.
Thank you, Michael Rinker, for the record. To conclude, cells respond to radiation exposure in a multitude of ways. The biological mechanisms discussed here depend on cell type, some of which have different radiosensitivities, radiation quality. As an example, high LET alpha radiation induces more complex types of DNA damage than low LET beta or gamma radiation, and biological endpoint. What was the observed or measured experimental outcome? Today, we've discussed DNA damage as an example in some detail. The balance between positive, negative, or neutral effects at the cellular level may or may not impact overall health. Given the outstanding uncertainties in the low-dose range, there is not enough scientifically sound evidence to warrant a change from the current radiation protection framework, which bases its dose limits found in the radiation protection regulations in part on the LNT dose-response model. Any uncertainties in risk estimates are further minimized by the use of the ALARA as low as reasonably achievable principle. Together, the dose limits and the ALARA principle effectively minimize worker and public exposures to safe levels. Given all of the information presented today, it is staff's conclusion that the current Canadian Radiation Protection Framework is robust and protects workers and members of the public. With regards to next steps, staff intend to publish a technical summary of this work in the peer-reviewed literature. In order to continue to regulate based on the best available science, CNSC staff will continue to monitor developments in the scientific literature. Staff have also identified areas where potential research projects could be initiated. For example, future studies should focus on how biological mechanisms can influence cancer development in humans. Notably, these studies should consider all biological mechanisms and not focus solely on one mechanism. And both in vivo and in vitro studies that elucidate the signaling pathways are also warranted. Thank you. That concludes our presentation today, and we are available to respond to any of your questions.
Thank you.